Glad to see you all back. And we will be working today finishing up our unit on genetics and also then starting to really look at DNA and what it's for in the cells, how it actually works. So that's something that's been kind of a roundabout path getting back to DNA after we first talked about it back around our chapter 3. But we'll be doing a lot with it in the remaining weeks. So we'll be doing also an extra credit assignment today. This is one looking at blood typing, and with this you can pick up as many as 4 points extra, which I think a lot of people would probably like to have. So we have that. I'm also going to tell you something here I think you'll appreciate. And I've decided with these major quizzes that we're doing during the quarter, not with the final exam, but with the 3 major quizzes, I'm going to drop the lowest score of those 3. So that is something that I often do anyway during a term. I didn't have that booked into our class structure, but I'm going to do it after all. So basically, if you really tanked on the last test, that score can go away as long as you do better on the next one. So throwing you that as kind of a lifeline, because I know a lot of people were really hurting on that last test. And I don't think that takes away the need for you to study very intensively for the remaining major tests we have. We have one more of these large quizzes and the final exam, which I'm not going to toss out. But I think that'll give you a little more of an edge in getting your grades up. So out of the 3 major quizzes, I will throw out the lowest of those 3 scores. Otherwise, we have a lot more work to do, and we should be getting on with it here today. So let's go and take a look at our Canvas. And I need to let someone in here. Let's see. Okay, so week 6 is where we are now, and I've done a lot of restructuring of the Canvas just now. One thing is I put in a link to the tutoring sketches that Highline has. So before the next major test, you may want to talk with the tutor. We have a tutor who's working remotely, but through the tutoring center 3 days a week. And I think before you take another major test, it might be in your interest to talk with that person. So I have the link here for all the tutoring schedules, which includes biology. I have then lecture materials here dealing with genetics and DNA. And this one, excuse me, that should say chapter 11. So let me alter that right now. Okay. So I have in here the lecture materials dealing with genetics and getting into DNA, and also practice problems from a different book dealing with these same topics. I have in here a link looking at blood typing, which is a thing that we'll be working with today. We'll be getting into that pretty quickly, and that's what your extra credit is on. I'd recommend after we do the extra credit assignment that you also go through this section with a biology project. Also some things about how you might be your own evil twin and not even realize it. And I have several links here to Khan Academy items dealing with what we're getting into this week. The homework for extra credit is right here, blood types, extra credit. And this is a thing I'll give class time on for us to work on today. And videos that we will be going through many of these, maybe not all of them this week, but many of these this week. So these are all things that I have in here now for our current week. It's quite a bit of stuff. But I think you'll find quite a bit of it to be pretty interesting. And do you have questions? Otherwise, I would like to start back in with genetics.
Is it possible to upload that blood typing extra credit as a Word document?
I can do that. So yeah, let me do that right now. Actually, let's wait until I'm actually turning you loose on it, and I'll upload it then. So otherwise, we have some things to do now before we get to that. And but I'll put that up there when we start with this assignment. Well, looking back at our genetics, I'm going to go back to about where we left off. We were looking at the Punnett squares, various things like that. We had these Punnett squares where, for instance, we had the idea that, and are you seeing now this PowerPoint screen?
No.
Okay, then I will bring that right up. Sorry. Here we go, PowerPoint. So we were looking at this very simple experiment involving breeding of peas that Gregor Mendel did. And the key thing about this is that it really disproved the idea that inheritance works by blending. You had here two different varieties of these flowers, and you ended up not getting the trait per flower blended. Instead, it looked as if these hybrids were exactly like one of the parents, and it looked as if this white characteristic, what we now know as recessive, had just vanished from the population, but it was still in there. It was in the hybrids. We knew that because when we bred hybrids with each other, some in the third generation came back white. That showed that essentially when we are inheriting information from both parents, we get information, genetic information from both parents in our cells, and those different, what I'm thinking of as particles of inheritance, can be passed on separately to our offspring. So in this case, the hybrids inherited something, we'll call it a particle of inheritance, from each parent. And so there were two of these particles of inheritance, genes, within the hybrids. And then when they made their own offspring, they passed these on separately to different offspring. The different offspring got one of the genes or the other one. They didn't get both. And it's because they could get, say, the white recessive copy of this gene from both parents that we end up getting a white offspring down here. So this demonstrated that you can have a gene hiding where you don't see its effect, and yet it's still there, and it gets passed on through the generations. That was really key to understanding how genetics works. So this experiment was extremely important. Now, as we're going to start to see, real genetics gets a lot more complicated, but essentially the foundation for understanding it starts right here. So we went through this. We ended up predicting then, that is, Mendel ended up using that model, predicting that you'd get about a 3-to-1 ratio in the offspring when you're comparing the purples and the whites in this third generation. And in fact, that's essentially what he got. He had this sort of result when he was doing the flower color, approximately 3-to-1, very close to 3-to-1, close enough that it supports his model. And he did this for all the other characteristics he looked at, for things like size of the plant, whether it was normal or dwarf, whether the peas were yellow or green. He had several other types of traits, and they were all inherited in the same way. And he ended up with approximately 3-to-1 ratios on all of those. So very convincing results. Well, I'm going to come back to this dihybrid cross later. Otherwise, I would like to start us into blood typing right now. So I'm going to skip this little part and come back to it in a bit. So here, blood types. When we're talking about blood types, what are the typical types that we hear about people having? What sort of things come to mind?
A, B,
A,
Positive.
O,
Yeah.
and then the positives.
O, positive, and so forth. Right now, we're not going to talk about the positive or negative. What we have is one gene that controls the ABO types. That means types A, B, AB, and O. One gene that has 3 different versions instead of 2 different versions controls whether you have any of those 4 blood types. There is a different gene, which we're not going to talk about yet, that controls whether it is what we call positive or negative. Right now, we're going to ignore the Rh gene that gives you positive or negative. Let's just look at the ABO gene. This is getting more complicated than the flower color situation. There we only had 2 versions of a gene you either had the white or the purple version. But real genes often have more complexity, more variations. So what we have are three different types, three different alleles, three different versions of this gene. And what happens with these different versions is they code for antigens. Those are surface markers that go on the red blood cells, and they are identification tags, essentially, and they can trigger an immune attack. What will happen is that you may end up having none of these antigens, in which case you actually are type O, or you might have one of these antigens, which means you're either type A or type B, or you might have both antigens, in which case you're type AB. But these antigens are markers which can trigger an immune attack. Now, later, when you're in anatomy, if you study the immune system, you'd see all about antigens, much more than you want to know, really. But we're talking here about a single gene which, at most, can code for two different antigens, triggers that could trigger an immune response attacking the blood. So these are things which can trigger an immune response. The thing is that if you already are one of these blood types, let's say you already are type A, then you're making the A antigen, but your immune system doesn't attack your own blood. That is something where your immune system is trained normally not to attack your own tissues. Now, we sometimes will get autoimmune diseases where people do have some localized situation where their immune system attacks their own tissues. But in general, people don't have those. And so if you are type A blood, you make type A blood, your immune system is not going to attack your type A blood. And it won't attack anyone else's type A blood if it's transfused into you. But if you are maybe type O and you're not making any of these antigens, then if they go and transfuse type A blood into you, your immune system will attack it as being foreign. Your immune system will think that you're being invaded by bacteria and will try to destroy this foreign blood. So there are a number of different ways this can happen, where a person with one particular blood type may or may not accept other transfused blood, and that is something we're going to be looking at. We'll look at the details on this more in a second, but are you with me at this point on this idea?
Yes. Yeah, quick question real quick. Is this type O blood doesn't make sense? That's why it can be a universal donor?
I was having trouble hearing you. I'm going to turn up the volume a bit.
Because type O blood is doesn't have any antigens, is that why it can be the universal donor?
Yes. Yes, exactly right. Type O blood basically will not upset anyone's immune system. Type A blood would be more restrictive in terms of who can get it. So, yeah, you're exactly right on that. Well, we'll see more with this. So, what will happen is that if they are checking, let's say in a hospital, checking what your blood type is, maybe you need to get a transfusion because you've lost a lot of blood. Well, they don't want to put the wrong blood into you because that might be worse than not doing it at all. If they put a couple of units, pints of blood into you, and it's the wrong type, that could kill you. So it's worth, let's say, checking out your blood type before doing any such transfusion. And that's very easy to do. I've done it. And where all you do is you have a little bit of a well, just a small, you got a little plastic tray that's about this big, and it has little indentations in it, and you put drops of the person's blood into those little indentations. And what you'll do is you put in what's called an anti-A serum. It will react with type A blood and cause the blood to clump together, which is what's happening over here on the right. Notice what happens here at the lower left. We have in our bloodstream antibodies. These are defensive proteins that will stick to things like viruses and bacteria and cause them to become agglutinated, many of them stuck together into masses. It neutralizes them as far as being harmful invaders and makes it easier for a white blood cell to come along and just gobble them all up and destroy them. So the antibodies will stick to various foreign antigens that are on the surfaces of things. If we transfuse the wrong blood into a person, antibodies will do the same thing and cause all those red blood cells that we transfuse to stick together in these masses, which can plug up your blood vessels. But we can see easily what happens with that. If we have blood and it stays unagglutinated, where it didn't react against the anti-A serum, it stays spread out like we get here in the middle. But if we put the blood in with, say, anti-A serum, and it is type A blood, then the blood will stick together, as we're seeing at the left. And that would indicate this person is type A or AB, either one. It has an A character to it. So well, we can do this easily in just a few minutes to test what someone's blood type is. So what happens is that we have 3 different versions of this ABO gene, and we write them as you see here at the left. And when you're working on this activity, you must write these out this way. Don't try to do a shorthand like just writing an A or a B. Very, very clear on this. When you're turning in this assignment, if you want credit on it, then any place it says to write an allele, you must write them exactly as I've given here. Capital I with a capital A, capital I with a capital B, or lowercase I. So we're using the capital and lowercase again, big and little, to indicate dominant and recessive. But in this case, there's 2 different dominant versions. And what happens is that one of those dominant ones, say the capital IA, would cause this reaction that we see on the right here with the anti-A serum. The other one would cause this reaction with the anti-B serum. So we have 2 different antigens that can be on the red blood cells, and both of them can trigger this immune response. The lowercase I, little I, is recessive. It doesn't create any antigen on the surface of the red blood cells. Therefore, the immune system will not attack these, will not attack the little I. So any person can only inherit 1 or 2 of these. You might get 2 identical alleles or 2 different ones, but no one has a 3rd copy because they would have to inherit 3 copies of this chromosome. I think it's chromosome 7 that has this, and there's nobody alive in the world who has 3 copies of chromosome 7. So at most, a person only gets 1 or 2 of these, never all 3. So there are 6 different possible combinations that show up. Here are the different genotypes that people can have. A genotype is a description of the genes when you're focusing on a particular gene. This is giving us a visibility on what the different kinds of genotypes are regarding this gene. People could be pure type A or AB, or a different kind of type A where they have this recessive little I along with the dominant big A, big IA, or they can be B 2 different ways, or they can have this last one. What blood type is that last one?
O.
Yeah, that's O. So what we have here again, reading across the blood types, which are the phenotypes. When we see a phenotype, well, that is actually what we see. Like a purple flower is an example of a phenotype. With the flower color, if we said big P, that was a genotype description of a purple flower. Here, these are genotypes, the underlying genetics that tell us how we get the different phenotypes, which are the blood types that we speak of. Type A, AB, A again, B, B, and O. So when we do the transfusions, if a person already makes the IA antigen, their immune system is trained not to attack that antigen. You have self-compatibility. Your immune system should not attack your own tissues. So it goes through an education process and learns basically which tissues belong, which antigens belong in your body and which ones don't. So in this case, if you are already making the IA antigen, you're not going to attack the blood that has that. But there's more to it than that. Similar idea, if you're already making the IB antigen, you won't attack that if it's in transfused blood. But there is a trick with that. What if you are type A and you receive AB blood? What happens then?
Then you will attack the B component.
Yes. Yeah. Your immune system wouldn't care about the A part, but it would attack the B fraction. So that's a reject. If you are pure type A or any kind of type A or type B, you cannot receive AB blood because your immune system will attack half of it. And that's as bad as a full attack. But if you were type A and you were receiving type A blood, no problem. And again, the little I allele doesn't produce any antigen to trigger an immune attack. So, who can donate blood to whom? Well, this is one thing I'm going to have you work on when you're doing this assignment, which I think you could start with right now. So, let's go back to where I have the worksheet for you, and I'm going to go uploading then the Word formatted copy of this for people preferring that. And where you get this assignment, it's going to be down here under week 6 homework. Anybody wanting to download that as a PDF can do it right now, and I'll have the Word formatted one up there in a moment. And then I'm going to break you out into groups and give you some time working on this. Right, so blood types is there, and I'll go in the other section and do the same thing. So this one's for extra credit. Oh, sorry, not that. Wrong thing. So I have the PDF and .doc versions of this here for you. And so you should be able to then see this under our week 6. You'll find that in there under homework. Blood type, extra credit, blood types.doc, they're the same thing. So everybody should be getting a copy of this, looking at it, seeing what you can do. Now, on one side, it asks you what which blood types are accepted or rejected by people. So across the top, it's going to be on this matrix. It's going to be saying, here's the donor. Down the left, it's going to say, here's the recipient. Who can give blood to whom? In fact, I will bring that up so we can look at this a little bit. right, so here's what the assignment looks like. I have a due date on there of 7:30. Ignore that. I thought I might give this to you last week when I first formatted the file. I'll give you a corrected place to turn this in. But I'm asking you to give me what the genotypes are. And with that, you're having to write 2 alleles. Remember, there's 3 alleles, and each allele needs to be written in exactly the formats that we see here. Any one person inherits some combination of 2 of these. You just write them together. You don't have to separate them with a comma or anything. You can just write like little I as an example of a genotype. But I want you to get the 6 different genotypes here, telling me what the different blood types are that go with those. And then here we have the donor to recipient matrix. We need to see whose blood is accepted by whom. And what you want to do is write either an A or an R in each of these blanks. I will tell you there are 7 rejects. 7 rejects, 9 acceptances. 2 questions here about who can receive blood from anyone, who can give blood to anyone. And then here on the back, this is kind of fun. This is deciding who's the daddy, which is always kind of an entertaining thing to do. For a long time, when it comes to deciding whether someone owed child support because of maybe getting a girl pregnant and then trying not to take responsibility for it, courts relied on blood types as a way to tell who the father might be. And so if you had, say, a woman who had gotten pregnant, if that wasn't, say, married, she said this boyfriend had gotten her pregnant, and now he owes her for child support, and he would be insisting, no, it's not my kid, it must have been some other guy's. Well, they would do blood tests and see whether or not the blood types were compatible for the guy actually being the father, and whether that would give support to what the woman was saying. Or maybe it would turn out the blood types weren't compatible, and the guy was actually telling the truth. He wasn't the father. So here I give you 3 cases where we know what the baby's blood type is, and you can give a genotype in these blanks. Again, you're using genotypes as we've already described. Just use as many blanks as you need. With type O, there's only one way to be a type O, so you don't need to use both blanks there. Type A, well, there's 2 ways to be a type A, so you use both of those. The accused father is AB. We already know what the baby's blood type is, and the mother's. The question is the father. We put down the accused father, and we see whether he could actually be the father or not, and then do the same thing again and again. You're deciding whether the guy could actually be the father. So this is a thing that's actually kind of fun to do, and I want you working in some groups here, and I'm going to break you out into groups right now to work on these. Then we're going to come back, and we'll be in an emergency room dealing with several accident victims and seeing what kind of blood we can give to them. So, are you ready to start with this? Okay, breaking you up into 7 groups then.